water in the southwestern region of the U.S. So, some background information on Cyclospora is it's a coccidioporozoan parasite which causes gastrointestinal illnesses in humans called cyclosporiasis. The oocysts reproduce in the lower GI tract of humans. It can perform asexual and sexual reproductions in the intestine of the infected individuals, which will be then shedded in the infected individual's feces for up to several months without treatment. The life cycle continues in the environment. It takes 7 to 15 days for the oocysts to sporulate and become infected. I've included a picture of the life cycle of Cyclospora and how the oocysts sporulate and how they survive in the environment. Over here to the right-hand side, this is the life cycle of Cyclospora on the outside of the environment and before and after it is sporulated. This presented study assesses the prevalence of Cyclospora cayetanensis in agricultural water in Yuma Valley fresh produce growing regions by collecting monthly samples up to 100 liters of agriculture samples for a total year of 196 samples. Cyclospora cayetanensis was collected from EnviroCheck high volume filters. The processing method that we used was the EPA method 1623. The newly developed FDA BAM19 BIM method was used for the DNA extraction and the DNA purification. We used the quantitative PCR for our detection method. A total of 3% of the samples were positive for Cyclospora cayetanensis and were detected during the winter months of December, January, and February, and all their samples were negative. Example of produce that have been confirmed to be infected with Cyclospora cayetanensis include cilantro, raspberry, and basils. As of August 28th of 2019, the CDC reported that there have been about 1,696 laboratory confirmed cases of cyclosporiasis. In 2018, there were 2 major outbreaks that occurred that infected 250 reported individuals. All these individuals were infected by the consumption of salad or vegetable trays that were contaminated. The most recent reported outbreak was September of 2019, where there was a total of 241 confirmed reported cases. These cases were traced back to the consumption of contaminated fresh basil in 2019. Here in my method sections, I have how we collect the sample out in the field. We had a pump that would suck the water up from the canal through a hose into an EnviroCheck filter. The EnviroCheck filter had filters inside with pores inside that would capture any cyclospora or any other bacteria that would be caught in the pore itself. Then the water would be pushed through the other side of the hose and be put back into the water canal, and we had a meter attached to the end of that side of the hose. That is the way we were able to keep track of how many liters of water we filtered through the EnviroCheck filter. Next, for the processing sample, we used the EPA method 1623 to process our samples, and we used the FDA BAM19B method for our DNA extraction, which is pictured here, with our DNA purification, which is also pictured there. Our detection method using our qPCR, which our qPCR then leads to our results section. In our results section, I have included the months that were positive and whether those canals were lined or unlined canals. Underneath that, I have also included the standard curve on what we would look for in our results after every single one of our runs. Our standard curve is our positive control, and it also checks the efficiency of our run. That's something that we looked at every single time on every single month of our samples. I have also included an amplification curve to show for how long we would run our qPCR cycles for, and the CT value that we would use is 38. So anything above 38 was considered a negative. Anything below was 38 was considered positive. Some discussion and conclusions. These findings suggest that the risk of Cyclospora cayetanensis and fresh produce contamination from irrigation water in this produce growing region seems to be relatively low, given that no Cyclospora cayetanensis outbreak has been associated with fresh produce growing in the Yuma Valley area, and given the volume of raw agricultural irrigation with these agricultural waters. The recommendation is for the irrigation districts and growers to continue to implement and practice current water quality safety measures and good agricultural practices and good management practices to continue to reduce the potential contamination of fresh produce with agricultural water contaminated Cyclospora cayetanensis oocyst. That is the end of my presentation. Thank you so much.